This video is Chapter 28, Notes Part 2, all about what occurs at the synapse, meaning the space between two neurons. So to remind you, in the previous set of notes, we learned that a neuron has some dendrites, and those dendrites are what receive a signal. The signal could be sensory input, such as sight or taste or sound, or it could be a particular molecule. Generally, the AP Bio questions focus on the input being a particular molecule. So that molecule is going to end up binding to the dendrite. Upon binding to the dendrite, the action potential gets sent down the axon. The action potential, to remind you, is not a molecule moving through the neuron, but rather it is a movement of ions. So essentially, when the neurotransmitter binds to the dendrite, the first set of sodium channels open. Sodium then flows into the neuron, and as the first set of sodium channels open, it causes a domino effect for each neighboring sodium channel to then open. Eventually, you reach the end of the axon, at which point there is the terminal, goes by a couple different names, terminal buttons. And at the terminal then, a new neurotransmitter is released. So this video will go through all of that. First, the location where we're focusing in this set of notes is called the synaptic cleft. It's the space between two neurons, meaning these neurons here are not physically touching. They are not fused together. There is a teeny tiny microscopic space between them. Therefore, the molecule, meaning the neurotransmitter, is going to have to travel through that teeny tiny space. The terminal is the end of the neuron, so the terminal would be this area here, the end of the neuron, and the end of the neuron contains vesicles that are filled with neurotransmitters. I think of a vesicle almost like a little Ziploc baggie filled with the neurotransmitters. As you can see here on this slide, these are the vesicles. So they're like little Ziploc baggies filled with the neurotransmitter. Those vesicles will only be released when an action potential is sent. Neurons are not constantly talking to each other. They only talk to each other when needed, meaning when there is some kind of input and the cells have to communicate with each other. So when a neuron is at rest, these vesicles here are just stored in that terminal. Calcium is going to be a big player for us today. Calcium is another ion which is typically on the outside of the neuron when the neuron is at rest. So when at rest, calcium is on the outside. When an action potential is sent, sodium channels sequentially open in the axon, meaning when one opens, its neighbor opens, its neighbor opens, and that's what I mean by like falling dominoes. The first channel has to open in order for any of those subsequent channels to open. When the last sodium channel opens, it then causes an ion channel to open, allowing calcium to come in. Calcium will not come in until that last sodium channel opens and tells calcium essentially it's okay to come in. So when that last sodium channel opens, it causes the calcium channel to open, allowing calcium to flow in to the terminal. Calcium then shoves those Ziploc baggies to the cell membrane. So calcium is going to cause those vesicles to fuse with the cell membrane here and essentially open up and spill out the neurotransmitter. So calcium enters when the last sodium channel opens. Calcium then causes those vesicles to move and fuse with the cell membrane. When the vesicle touches the cell membrane here and those vesicles fuse, it causes the neurotransmitter to be released then into the space. So the neurotransmitter would then be in this space between the two neurons. That neighboring cell has a dendrite. Remember, dendrites are where the neurotransmitters bind. So that neighboring neuron has a dendrite that contains receptors. A receptor is a protein with a specific shape, and that receptor protein can bind that neurotransmitter. When that neurotransmitter binds to the receptor, it will then cause a sodium channel on that new neuron to open. Those sodium channels would sequentially open. Eventually, over here-ish, we would reach the terminal of that second neuron, where we would again see the last sodium channel open, allowing calcium to come in, moving the vesicle, releasing the neurotransmitter, and this process would occur over however long of a distance the neurons need to communicate. That neurotransmitter is a molecule used as a signal. So that neurotransmitter that's normally stored in the vesicles is going to act as a signal to start the action potential in the neighboring neuron. So when the neurotransmitter binds to the dendrite, it causes the action potential to start in that neuron. As you can see, different neurotransmitters each contain different shapes. Those receptors on the dendrites each has a specific shape. So depending on which neurotransmitter binds, that would cause a different neuron potentially to respond, depending on the shape of the receptor that that neuron has. So when it comes to neurotransmitters, again, shape is important. We talk about shape a lot in regards to proteins. The receptor is a protein, it has a shape. The neurotransmitter is a molecule, it's not technically a protein, but it also has a particular shape. Those shapes need to fit together in order for the neurons to communicate. So all our knowledge about cooling and raising temperature would come into effect in regards to the effects it would have on the protein. If I were to make something very hot and I had an extremely high fever over 104 degrees, I better get myself to the ER because those proteins in my brain, those receptors on the neurons in my brain are potentially going to start to denature. Well, my brain controls everything, my heart, my diaphragm, which controls my lungs. So if those proteins in my brain denature, that is extremely bad. Similarly, if it gets too cold, the motion of that neurotransmitter is going to be so slow such that it might not bind to the receptor in a timely enough fashion. So again, we still will keep in mind all of our temperature ideas when it comes to proteins. This kind of reiterates at the top what I said before in a nice picture for you. So we have here the terminal of one neuron, and that neuron has an action potential that moves down the axon. That action potential is simply sodium channels opening, allowing sodium to come in, and then sodium immediately being pumped back out. When those last sodium channels open, I have a vesicle. That vesicle is going to move toward the membrane, but it requires calcium to do so. So when those last sodium channels open, calcium will come in and shove that vesicle toward the membrane. My neurotransmitter is then in the gap, and that neurotransmitter is going to bind to the receptor proteins. Shape is critical. Only some neurotransmitters can bind to some receptors. When that neurotransmitter binds, it will initiate an action potential in this neighboring neuron. At some point, the topmost neuron would potentially run out of neurotransmitters because it's spilling them all into the gap. And then that neuron would be totally useless. So that does not happen. Instead, there is an enzyme, and that enzyme cleans up the gap. So it essentially takes those neurotransmitters and it repackages them back into the vesicle so that that neuron could fire for your entire life. Your neurons really don't replicate much, so one neuron lasts pretty much your entire life. So you repackage those neurotransmitters so that the neuron can fire again. And there's an enzyme that does that. This is the same picture. So to conclude, there's a few ways that that normal neurotransmitter pathway can get screwed up. So we're going to look at a few examples of those, and there tends to be a lot of College Board questions about how things get screwed up. So we'll take a look at a few examples. It's not important that you memorize which drug does what, but more so you think about if this is this type of drug, what impact would that have? So here our first example is heroin, which is an opioid. So all opioids work in a similar fashion. The way that an opioid works as a painkiller is that it prevents communication between two neurons such that the receiving neuron never knows that there's a sensation of pain. So shown here in this picture, I have an action potential being sent down a neuron. That action potential should result in calcium coming in to the terminal and moving the vesicle so that the neurotransmitter is released into the gap. That does occur as normal. However, opioids work by blocking receptors on neighboring neurons. So my neuron down here at the bottom should be receiving a signal which will eventually go to my brain telling me about the sensation of pain. However, the receptors on that neuron are blocked. The heroin molecule or the opioid molecule binds to those receptors and prevents the normal neurotransmitter from binding. Therefore, the neighboring neuron never receives a signal and will never send an action potential because the neurotransmitter is not binding. So any chemical that works to block the receptor would prevent the action potential in the neighboring neuron. Simply because shape is important, the drug is blocking the shape of the receptor, so the neurotransmitter cannot bind. Snake venom, in some cases, works that same way. It acts as a neurotransmitter block. A snake uses venom to hunt and kill prey. The snake wants to paralyze its prey so that the prey cannot escape. So the way that the venom works to paralyze the prey is by preventing neurons communicating with each other and preventing neurons from communicating with muscles so that the prey animal cannot move away. Again, the receptors on the neighboring neuron or on the neighboring muscle would become blocked with a chemical that is similar in shape to the neurotransmitter, preventing the neurotransmitter from binding. Another example of a neurotoxin would be one in which the gap is not cleaned up. Cocaine is an example of that. Cocaine is a stimulant, meaning it causes your neurons to fire when there is no actual sensation or no input, so thus you have like hallucinations. In chemicals like that, where the gap is not cleaned up, what happens is the neurotransmitter stays bound to the receptor longer than it should. So that then causes the action potential to be sent more frequently than it naturally should. So essentially, the presynaptic neuron, so this neuron here at the top, is not firing, but yet the postsynaptic neuron down here is firing. So these neurons are not actually communicating with each other. The bottom neuron is firing when it should not be. And the reason for that is because cocaine, which is similarly shaped to a naturally occurring neurotransmitter, there's my natural neurotransmitter, cocaine is similarly shaped. So cocaine binds to the receptor due to it having the same shape, causing the bottom neuron to fire an action potential when it really should not be. Another example that's similar also causes stimulation in the neighboring neuron, meaning it also causes an action potential in the neighboring neuron when it should not. And that's like hallucinogens, like some varieties of mushrooms allow people to hallucinate and see things or hear things that aren't actually there. And in that case, we don't have the gap being cleaned up, but rather there's no action potential being sent, yet the neurotransmitter is still being released. So again, with all of these examples, you want to figure out, is this going to prevent an action potential, or would this cause an action potential? Another example of a neurotoxin is in a pufferfish, which creates a poison to prevent being eaten by predators. And this particular neurotoxin works by blocking the sodium channels. So normally we should have sodium that flows into the axon, allowing the neuron to send an action potential. So those sodium channels should be opening, allowing sodium to come in, but in the case of the pufferfish, it produces a toxin called tetrodotoxin, which binds to the sodium channels, and thus sodium cannot get in to the neuron. So if sodium is not getting in, I would have no action potential. Thus, all of my neurons downstream would not receive a signal because this neuron is not sending an action potential. Few last examples. Tetanus is another example. You probably all have received a tetanus shot. You get one about every 10 years. Tetanus is a bacteria that lives naturally outside. If it gets into your bloodstream, it can cause what they call lockjaw, which essentially means that your muscles contract and they do not relax. That's especially bad if you're thinking of a muscle like your diaphragm or your heart that controls normal body functioning. But in tetanus, what we have is that the vesicle is not releasing the neurotransmitter. So here we have an example where if there is no neurotransmitter, the neighboring neurons will not respond. Another example from a poisonous snail is blocking the calcium channels. So if given a scenario where the calcium channel is blocked, you should understand, well, if calcium is not coming in, my vesicle is not moving, and my neurotransmitter thus will not be released. So again, with all of these, think big picture. Are we sending an action potential, or are we preventing an action potential?